Hi, my name is Jessica Tuohy. I am a scientific product manager for the DiaPharma group, and today I'm going to talk about cytokeratin 18 or CK18 as a non-invasive biomarker in NASH clinical trials. Non-alcoholic fatty liver disease, or NAFLD, is an umbrella term that refers to a spectrum of conditions where there is accumulation of excess fat in the liver of people who drink little or no alcohol. It is a progressive disease that starts with relatively benign fatty liver or steatosis. About 20% of individuals with NAFLD develop non-alcoholic steatohepatitis, or NASH, which is a more aggressive form of the disease. NASH is characterized by liver inflammation, hepatocellular damage, and fibrosis. Some people may progress even further to develop cirrhosis and hepatocellular carcinoma or liver cancer. At that point, a liver transplant is generally needed for survival. While the risk of liver-related morbidity and mortality is significantly increased in NAFLD, complications from cardiovascular disease are actually the leading cause of death in this population. During the past 20 years, the incidence of NAFLD has more than doubled, and it is now the most common liver disease in developed countries. In the United States, about 80 million people, or a quarter of the population, have fatty liver disease and are on the NAFLD spectrum. Over the next 10 years, these numbers are expected to further increase to over 120 million. And even more alarmingly, 10% of the pediatric population in the US is thought to have NAFLD. The increased incidence in NAFLD is strongly connected to the epidemic increase in type 2 diabetes and obesity. And this year, NASH is projected to overtake hepatitis C as the leading cause for liver transplants in the US. Unfortunately, physical symptoms are often silent or nonspecific to NASH, which makes it difficult to diagnose. As a result, individuals with NASH can remain unaware of their condition until late stages of the disease. I should point out here that not everyone with NAFLD will progress to develop cirrhosis or liver cancer. And in addition, NAFLD is generally a very slowly progressing disease, and it takes years or even decades to progress from steatosis to cirrhosis. And I'll come back to that point in just a couple of slides. NAFLD is a complex and multifactorial disease. NASH progression results from numerous events originating within the liver, as well as from signals derived from the adipose tissue and the gastrointestinal tract. There is now a consensus for a multi-hit theory that suggests that NASH is the consequence of several insults acting in parallel in addition to hepatic lipid accumulation to cause disease progression. These include genetic factors, insulin resistance, adipose tissue inflammation, ER stress, hepatocyte injury and apoptosis, activation of hepatic stellate cells, and microbiota dysbiosis. So even though we now have a pretty good understanding of the pathogenesis of NASH, there are currently no FDA-approved pharmacologic therapies. Insulin sensitizing drugs such as pioglitazone and metformin, as well as vitamin E, have been evaluated as treatments for NASH, but each has drawbacks and limitations. Weight loss through diet and exercise remains the recommended treatment strategy for NAFLD, but unfortunately, only a small fraction of individuals is able to achieve and sustain therapeutic goals. So there's very clearly an urgent need for pharmacological treatment options for this growing population. And the field of companies that are in the process of developing NASH drugs is wide. The complex etiology of the disease offers many therapeutic targets, which is reflected in the multitude of agents that are currently under investigation. The main categories of drugs that are in development include those that affect the various metabolic pathways, so de novo lipogenesis and bile acid circulation, hepatic insulin resistance, apoptosis, oxidative stress and inflammation, and mechanisms impacting fibrosis. The number of trials for NAFLD and NASH that are initiated each year has been increasing exponentially, and there are currently around 8 80 active NASH trials underway. The first drug with a NASH therapeutic indication, obeticholic acid or Ocaliva, is expected to get conditional approval by the FDA sometime this year. But there are many more drugs with promising clinical trials data in development. So as you just learned, NAFLD is a very slow progressing disease. Unfortunately, this makes it virtually impossible to use hard clinical outcomes such as progression to cirrhosis, liver cancer, or mortality as endpoints for drug approval. That is because a large number of subjects would need to be followed for a decade or more, and that's just not feasible, even just from a financial standpoint. So that has led to the use of surrogate endpoints that are predictive of clinical benefit for accelerated approval in the US and conditional approval in Europe. Both the Food and Drug Administration, or FDA, and its European counterpart, EMA, are now accepting changes in liver histology as surrogate endpoints to attest efficacy of therapeutic treatment in NASH clinical trials. The histological response can be evaluated by a semi-quantitative scoring system that has been validated by the NASH Clinical Research Network, or NASH-CRN. It can be divided into two categories, the NAFLD activity score and the NASH fibrosis stage. Both of these are determined by a histopathologist's examination of a liver biopsy sample. The NAFLD activity score, or NAS, is a composite of three values assigned to the extent of steatosis, lobular inflammation, and hepatocyte ballooning that are observed in a liver biopsy sample. Each component is given an individual score, and the NAS is the sum of the three scores. Values range from 0 to 8, and the higher the score, the more severe the disease. The NASH fibrosis stages range from F0, meaning no evidence of fibrosis, to F4, indicative of cirrhosis. And one analogy that I've heard to relate the NAS and fibrosis stage is that of a road trip. The fibrosis stage tells you where you are in your journey, and the NAS tells you how fast you are traveling along your route. The surrogate endpoints that are now accepted by the US and European regulatory authorities for registration are either the resolution of NASH without worsening of fibrosis, an improvement in fibrosis without worsening of NASH, or both. The resolution of NASH is defined as the absence of ballooning, so a ballooning score of 0, and little or no lobular inflammation, so a score of 0 or 1. Improvement in fibrosis is defined as an improvement in the NASH fibrosis stage of 1 or greater. To be sure, after the accelerated or conditional approval, pharmaceutical companies are still required to do post-market monitoring of clinical outcomes in order to get full approval for their drug. It is important to note that the histological surrogate endpoints that I just talked about still require a liver biopsy sample to be taken. But biopsies have several drawbacks. They're invasive, costly, there is a rare but not insignificant risk of complications, and they're inherently prone to sampling errors. In addition, they are not ideal, especially for early phase trials, since significant changes in histology may take some time to manifest, or at least longer than the duration of these early phase trials. Furthermore, because biopsies can be painful, study subjects are often reluctant to have multiple biopsies done in a relatively short amount of time, as would be required in early phase trials. Because of these limitations, there is mounting interest in identifying non-invasive biomarkers that reflect treatment effect at early time points and that are also predictive of changes in histology later on. So essentially, what is needed are non-invasive surrogate markers for these histological surrogate endpoints. Imaging tools such as ultrasound, MRI, and MRE have been used for assessing steatosis and fibrosis, and specific biomarkers, as well as panels of biomarkers, lipidomics, and metabolomics have been used to monitor changes in other NASH parameters, such as inflammation, oxidative stress, and apoptosis. The advantage of these non-invasive tests is that they can be done repetitively over a short period of time. However, I should mention that as of now, none of these have been validated by definitive trials to predict clinical outcomes. Okay, so now I'm going to switch gears for a minute and talk about hepatocyte cell death and its role in NASH. Then I'm going to introduce you to cytokeratin 18, and then I'm going to lasso those two together into a couple of biomarker assays and talk about their utility in NASH clinical trials. So hepatocyte cell death in the liver occurs primarily through one of two modes, either necrosis or apoptosis. Necrosis is an accidental form of cell death that occurs spontaneously in response to external factors such as infection or trauma. It results in the loss of cell membrane integrity, and the cell just kind of dumps out its contents. Apoptosis, on the other hand, is a highly organized and regulated process. Apoptosis is another term for programmed cell death or cell suicide. It involves complex receptor-mediated or organelle-initiated signaling pathways. Some characteristic features of apoptosis include membrane blebbing, DNA fragmentation, and cell shrinkage. Apoptosis also triggers activation of caspases, which are protease enzymes, and I will get back to those on the next slide. In 2003, Feldstein et al. Observed that hepatocyte apoptosis is significantly increased in individuals with NASH, and that the extent of apoptosis correlates with disease severity. Hepatocyte apoptosis is now widely recognized as one of the hallmarks of NASH. One of the major contributing factors to hepatocyte apoptosis is the excessive accumulation of lipids in the liver. This causes endoplasmic reticulum or ER stress and mitochondrial dysfunction, and eventually results in hepatocyte apoptosis. Apoptotic hepatocytes, in turn, activate immune cells and hepatic stellate cells, which leads to chronic inflammation, liver injury, and fibrosis. Hepatocyte apoptosis is thought to be an important, if not critical, driver in the progression of NAFLD and the development of fibrosis. Okay, so now on to cytokeratins. Cytokeratins, also known as keratins, are intermediate filaments. They are part of the cell cytoskeleton, and they support the cell structure. They are also thought to have a cytoprotective function, and they allow cells to adapt to stresses via various defense mechanisms. In fact, inherited mutations in cytokeratins have been linked to over 60 different diseases, which supports their essential cytoprotective function. One of the members of the cytokeratin family, cytokeratin 18 or CK18, is expressed primarily in cells of epithelial origin. In the liver, CK18 is highly abundant in hepatocytes and cholangiocytes, which are the epithelial cells that line the bile ducts. As I mentioned previously, steatosis, inflammation, and hepatocellular ballooning are key histological features of NASH. Ballooned hepatocytes are typically 2 to 3 times the size of adjacent cells, and ballooning is caused by injury to the cytoskeleton, and it has been shown to be accompanied by a loss of CK18 staining in the cytoplasm. CK18 is also a major component of Mallory-Denk bodies, which are inclusions that are made up of damaged intermediate filaments and that are found in the cytoplasm of hepatocytes. Mallory-Denk bodies have been observed in various liver diseases. As I mentioned on the last slide, apoptosis is accompanied by activation of caspases. These caspases are proteolytic enzymes that recognize a specific amino acid sequence on CK18, and it is cleaved into small fragments primarily by caspase 9, but also by caspases 3 and 7. When a hepatocyte dies, either by apoptosis or necrosis, its plasma membrane disintegrates and its cellular components, including CK18 and CK18 fragments, are released into the bloodstream. So CK18 levels in the blood correlate with hepatocyte cell death, and both intact CK18 and CK18 fragments have been used as serum biomarkers for hepatocyte necrosis and apoptosis. CK18 and its fragments can be measured in serum and plasma samples with standard immunoassays. These assays come in 2 varieties. The M65 assays include the M65 ELISA and the M65 EpiDeath ELISA. These assays are based on two antibodies named M5 and M6, and hence the name M65. These antibodies recognize all forms of CK18, so both intact CK18 that is released from necrotic cells, as well as CK18 fragments that are generated during apoptosis. The M65 assays therefore measure total hepatocyte cell death, so both apoptosis and necrosis. In contrast, the M30 ELISAs are specific for apoptosis only. The M30 assays include the M30 apoptosens and the M30 cytodeath kits. The unique M30 antibody in these kits specifically recognizes a neoepitope on CK18 that is formed after the protein is cleaved by caspases. So it only detects the caspase cleaved CK18 fragments and is therefore only measures apoptosis. So again, just to review, M65 kits measure both apoptosis and necrosis, and the M30 kits measure specifically apoptosis. The two kits can also be used in combination to discriminate between the two cell death modes. If the M65 levels in a sample is much higher than the M30 levels, which shown on the left, it indicates that the primary cell death mode is necrosis. However, if the ratio of the two is closer to 1, shown on the right, it is an indication that apoptosis is the predominant cell death mode. Since apoptosis is such a prominent feature of NASH, CK18 has been extensively studied as a biomarker for NASH and specifically for monitoring the progression of NAFLD. Several research studies have looked at a correlation between serum CK18 levels and NAFLD or NASH. Total serum CK18 and the caspase cleaved CK18 fragment have been shown to be increased in both children and adults with NASH compared to healthy individuals or those with simple steatosis. Since apoptosis is thought to be one of the drivers in the progression of NAFLD, elevations of serum CK18 levels may therefore represent active NASH. Serum CK18 has also been shown to correlate with each of the components of the NAS, so steatosis, lobular inflammation, and ballooning. And in at least 2 studies, changes in CK18 have shown stronger positive correlations with changes in the NAS than some of the conventional liver enzymes like alanine aminotransferase and serum aspartate, or ALT and AST. And even though CK18 levels are not a measure of liver fibrosis per se, CK18 levels have also been shown to correlate with fibrosis stage. So monitoring changes in CK18 levels in NASH clinical trials may be useful for monitoring treatment response and for assessing efficacy, especially in early phase trials before there's manifestation of histological changes. And in 2015, a joint workshop of the FDA and the American Association for the Study of Liver Diseases recommended a reduction in cell death markers, such as CK18, as secondary endpoints for NASH clinical trials. And CK18 has been widely used as a biomarker in clinical trials. The data on this slide are a bit outdated. They're from 2018. But at that point, CK18 had been evaluated as a biomarker in over 115 clinical trials and in 60 trials with a NAFLD or NASH indication. Most of these were early phase trials, so phase 1 or phase 2, but that's because at that point, there were only a few NASH drugs in phase 3 development. Since then, more NASH drugs have entered into phase 3, and we know that CK18 is included as a biomarker in the majority of phase 3 NASH trials, so we would expect that slice of the pie to be slightly larger at this point. Shown on this slide is just a small selection of early phase NASH trials that have used CK18 as a biomarker. This is certainly not meant to be an exhaustive list, but I just wanted to illustrate the variety of mechanisms of action of the different drugs, the different trial phases, and the differences in duration of the studies where CK18 has been applied. Some of the trials used both of the CK18 biomarkers, so both M30 and M65, and some only used one to assess either apoptosis or to capture total cell death. But in each of these studies, drug treatment caused a decrease of these biomarkers over baseline values. Statistically significant changes were observed as early as 6 weeks after treatment, which is much sooner than significant changes in histology would be expected. In a couple of studies, like the one for amricazan and DUR928, decreases in CK18 were seen within just hours of dosing, and this is likely due to a direct inhibitory effect of these drugs on caspase activity. So using CK18 might be useful as an early indicator of target engagement and drug efficacy in early phase NASH trials, keeping in mind that the observed time course is probably dependent on the mechanism of action of the particular drug that is being investigated. Changes in CK18 have also been shown to correlate with changes in liver histology. These are two older studies. They are phase 2 trials, and each one of them lasted 96 weeks. Phase 2 trials now are generally shorter. The PIVENS trial on the left evaluated the efficacy of pioglitazone or vitamin E in adults with NASH, whereas TONIC on the right evaluated metformin or vitamin E in children with NAFLD. Overall improvements in liver histology over baseline was a primary endpoint for PIVENS and a secondary endpoint for TONIC. The graphs in this slide don't actually show overall treatment effect, but rather subjects were stratified into those showing improvements in liver histology, which is represented by the open circles, and those showing no improvement, shown in the filled circles, and their CK18 values were correlated. And as you can see here, for both studies, individuals that had histological improvement also had greater decreases in CK18. I should probably mention that changes in ALT, which is another commonly used biomarker, also discriminated changes in liver histology in these studies. Here now are some more recent data from a phase trial that was conducted by Gilead to evaluate the safety and efficacy of selonsertib, a selective inhibitor of ASK1, either alone or in combination with simtuzumab in patients with NASH. Unfortunately, as you may already be aware of, last year the phase 3 trials for selonsertib, namely STELLAR 3 and STELLAR 4, were discontinued due to a lack of drug efficacy. However, selonsertib is also included in Gilead's ATLAS trial, which has looked at dual combinations of selonsertib with cyclophosphamide and fisorkostat, and I think Gilead is still in the process of evaluating how to proceed with that program. Regardless, in this phase 2 trial, selonsertib treatment alone had shown promising beneficial effects on both fibrosis and lobular inflammation. In the sub-analysis shown here, subjects were stratified into treatment responders and non-responders, similar to the last slide. Responders, so those that shows improvement in fibrosis or inflammation, are represented by the green bars, and non-responders, so subjects with no change or worsening of fibrosis or inflammation, are shown in red. For each group, the changes in CK18 levels were correlated, and the data showed that treatment responders for both fibrosis and inflammation also had greater reductions in both total CK18 and also CK18 fragments as compared to the non-responders. Like in the PIVENS and TONIC trials, these data support that changes in CK18 could be reflective of changes in liver histology. I want to emphasize at this point that we're not suggesting that CK18 alone could serve as a surrogate for histopathology in NASH trials, but rather using CK18 as a secondary endpoint could provide valuable early insight into target engagement and drug efficacy, including for NASH drugs with an anti-apoptotic mechanism of action. And it could therefore lend support in the decision-making process whether to proceed with further clinical studies. As I mentioned in the beginning of my talk, NASH is a complex disease with diverse etiology that will likely require a multi-targeted treatment approach, and therefore also a combination of biomarkers to get a complete picture of the therapeutic efficacy of the chosen treatment strategy. One more thing, for drugs with a purely direct antifibrotic mechanism of action, there are probably better biomarkers than CK18, since fibrosis is really downstream of the processes that affect and drive hepatocyte apoptosis. Finally, CK18 also has utility as a pre-screening tool for managing the screen fail rate in NASH clinical trials. One of the biggest challenges for NASH trials is the cost-effective and timely enrollment of study subjects. This is partly due to the large number of NASH trials that are actively recruiting subjects. As you may remember, there are currently over 80 trials underway. Adding to this is the difficulty of finding individuals with confirmed NASH. Despite NAFLD, there's an overall low awareness of the disease. As I mentioned in the beginning of the presentations, patients are often asymptomatic. And even those with a high likelihood of having NAFLD are rarely referred to a specialist, and the specialists are generally the investigators that participate in the NASH trials, so there's definitely a gap. In addition, subjects that have been recruited to participate in a treatment trial have to meet certain requirements to qualify for a study. Subjects are frequently required to have a NAS of 4 or greater and a fibrosis stage of F1 to F3. But more than 50% of screened subjects will not meet these eligibility criteria. So what happens is that these individuals will get a liver biopsy as part of the clinical trial, but then it turns out that they don't actually have NASH. Then the biopsy sample is examined, or at least they don't meet the inclusion criteria for the study enrollment. This high screen fail rate greatly adds to the cost and duration of NASH trials. One strategy to reduce the screen fail rate is to pre-screen recruited subjects with serum biomarkers and imaging tests prior to performing a biopsy, with the goal to enrich the study population that meets the required features for enrollment. As you learned earlier, CK18 is associated with hepatocellular ballooning, inflammation, and steatosis, which are all components of the NAS. So pre-screening subjects with CK18 assays and selecting those with high serum levels could increase the likelihood of these individuals meeting the required NAS threshold for inclusion. And this would then, of course, result in time and cost savings. So to review what I talked about today, NASH is a global epidemic. It is a slowly progressing chronic liver disease that can lead to cirrhosis, hepatocellular carcinoma, and other medical complications. There are currently no approved drugs for the treatment of NASH, but many clinical trials are underway to find an effective pharmacotherapy. One of the biggest challenges for NASH drug development is the selection of surrogate endpoints that predict clinical outcomes. Monitoring changes in serum CK18 levels may be useful for assessing treatment response, especially in early phase NASH studies. Improvements in CK18 correlate with improvements in liver histology, and using CK18 assays as a pre-screening tool prior to biopsy may decrease the screen fail rate in NASH clinical trials. So thank you so much for your time and your interest. Please don't hesitate to reach out to me directly if you have any questions or comments.